Hello, everyone, and welcome to the online version of the Neurotheory Forum, organized by Tim Vogels from IST Austria and Andrew Sachs from the University of Oxford, UK, and occasionally hosted by their colleagues like today. I am Everton Agnes, project leader at the Biozentrum, University of Basel in Switzerland, and today I have the pleasure to host Dr. Dan Goodman. Dan is a senior lecturer at the Imperial College London in the UK. And for those who are not familiar with the British system, a senior lecturer would be the equivalent of an associate professor in the US. Dan's background is in maths. In 2006, he received his PhD in mathematics from the University of Warwick in the UK, and then decided to switch fields to neuroscience. After a brief time at the Gatsby Computational Neuroscience Unit in London, he joined Romain Brett at the École Normale Supérieure Paris in France for a really fruitful postdoc. He later did a postdoc with Bertrand de Gut in Harvard before joining Imperial College London in 2014 as a lecturer. Let me see. And then has developed tools for both experimental and computational neuroscience, like spike sorting algorithms, in the experimental side, and spike neural network simulators, such as Brian in the computational side. And he's also the co-organizer of the NeuroMatch conference, which helped us continue our scientific interactions during this pandemic. But more importantly, Dan is helping us to understand how the brain functions and how it solves complex tasks. One important aspect of the brain is its huge diversity in terms of neurotypes and connections, and I really like Dan's approach to exploring the role of this diversity, which we'll hear a bit more today. So before we start, a few reminders. Use the chat as much as you want. If you have questions that you like to be answered during the talk, write them there. Additionally, there is an Ask Question box at the bottom of the page. Use it to post questions that you would like addressed at the end of the talk. And we will have a Zoom meeting with the speaker, Dan, after the talk for further discussions. I'll paste the link on the chat. So with no further ado, Dan, the online stage is yours. I will try to focus. Yeah, please.
Great, thank you very much. Thank you very much to the organizers, to Tim and Everton and everyone. My internet connection this morning was a little bit on the ropey side. It may happen that we have a few gaps where I disconnect for like 10, 20 seconds or something like that, but it seems to be coming back repeatedly. So if that happens, just hang on a minute. I'll be shortly back, assuming it hasn't got worse than it was this morning. All right, so let's leap into the talk. My title is Neural Heterogeneity Promotes Robust Learning. Let's get on to talking about that. The question of the talk is why is the brain so heterogeneous? And to sort of illustrate that a little bit, I just downloaded a few pictures of reconstructions of neurons from neuromorpho.org. If you don't know neuromorpho.org, it's a really cool website with thousands of cell reconstructions that you can download and play around with. So is this huge amount of diversity, and you see this wherever you look, and you see it in terms of the parameters of these neurons, and that's actually something that we'll be talking about later on in the talk, as well as their morphologies. So morphology is just a nice way of showing it, but you see it in terms of every aspect, in terms of which ion channels are being expressed to how much, and so on and so forth. So is all of this heterogeneity just some sort of quirk of the evolutionary history? Is it just developmental noise? Or is it playing some important role? And so that's the question that we were interested in getting at here. Okay, so that's what the brain is like. By contrast, if you look at our models, then they tend to look a bit more like this, right? Like particularly if you're looking at sort of models that I'm interested in, where it's models of a system that can actually carry out some sort of difficult information processing task. We tend to end up using entirely undifferentiated neurons. They're all the same type, they all have the same parameters, and it's only the connectivity that matters. So this is somehow what our models are like, and this is what the brain is like. All right, so before I get further on this, I just want to do a sort of little sort of side note on my philosophy of modeling the brain. As I was preparing this talk, I realized that the way I start this off, it might sound like I'm saying some things that I don't want to say. And so specifically, I don't want to say that just because the brain has a lot of detailed machinery, that means that in order to understand it, we need to reproduce all of that detailed machinery. And I'm not saying that our models have to be biophysically detailed or that they have to have connectivity guided by brain atlases or anything like that. Actually, quite the opposite, really. My view as a theorist is that in order to understand things, we have to simplify them as much as we can. And there's this very famous quote that everything should be made as simple as possible, but no simpler. And just as a little side note, this is commonly attributed to Albert Einstein, and it does originally come from Albert Einstein, but the actual wording is from a composer, in fact, Roger Sessions, in the New York Times in 1950. And you can see what Einstein actually said in the little box there. Okay, so the key part about this is that we have to make things as simple as possible, but no simpler than that. And so the question is, have we gone too far by making all of our neurons entirely the same, entirely undifferentiated? Is that an oversimplification or not? And if so, what did we miss with this oversimplification? What is the precise role that the heterogeneity we see in real neurons is actually playing? Okay. All right, so now let's do the 2-minute Twitter version of my talk. And so if you want, you can just listen to this slide and then you can phase out for the rest of the talk, because basically everything I'm going to say is going to be on this slide. So I'm going to start with the methods we looked at. So what the methods we used, sorry. So we're going to, what we looked at is heterogeneity of one very particular property. So there's all sorts of heterogeneity. And as I said in the picture before, we saw heterogeneity of the morphology of neurons. What we looked at specifically is heterogeneity of time constants. So that is like membrane time constants or synaptic time constants. So what I would mean by a homogeneous network is one where all of the neurons have precisely the same time constant. And if you look at modeling work, this is absolutely ubiquitous. Almost always you see that every single neuron has the same time constant, or maybe one or two different time constants, depending if it's an excitatory or an inhibitory neuron. But that's usually the extent of the heterogeneity that you see. So heterogeneity in our terms would be that every different neuron can have a different time constant. And that is more like what you see in natural neurons. If you measure time constants of a whole bunch of the same type of neuron, you see that they differ from neuron to neuron, often by quite substantial amounts. I've got some pictures of that I'll show you in a second. Okay, so I also said that we're interested in networks that can do complex information processing tasks, right? So what we did is we took spiking neural networks. From the intro, I hope you realize that I'm very much into spiking. Spiking is my thing. So it's spiking neural networks for me. We trained those to do difficult information processing tasks, like recognizing images or recognizing sounds, rather than very simplistic sort of laboratory stimuli. And in some of that training, we actually allow the time constants to be learned as part of the task. Now, we're not saying that that is necessarily a good model of how learning in the brain works. There may be some learning of time constants. We were just looking more for optimal distributions of time constants. So the learning is letting us find those optimal distributions. Okay, I think that will become clearer in a bit. All right, so now how about the results? Well, so some of our tasks had relatively little temporal structure. So we had some that are sort of visual static image tasks. They are basically, there's nothing changing over time. And what we found is that in those tasks, having all of this interesting heterogeneity of time constants basically didn't help at all. It didn't help solve the task. However, in those tasks which had more temporal structure, particularly the auditory tasks, having heterogeneity really helps improve the performance, improves the learning. I mean, I guess that's perhaps what you might expect, but it's nice to see that, and that gets a little bit towards the answer of the question: have we oversimplified in terms of making everything homogeneous? And the answer is, at least for some tasks, and definitely in the auditory system, probably the answer is yes there. Okay, Okay, so as well as finding that that helps, we found that the optimal distributions of time constants that we found actually look very much like what you see if you measure time constants experimentally. And so here's just a little figure of that. So here on the x-axis is time constant, I think ranging from about 0 to 100 milliseconds on a log scale. And you can see here that there's quite a wide distribution, and there's some like very large ones, and they tend to be sort of centered around some sort of mode like this. Okay, so that's what you observe experimentally. And this is what our simulations come up with. This is an optimal distribution for one of the tasks. And you can see that these are looking fairly similar. We see that there's the same like grouping mostly around some key value, but with a few values that are much larger than that. And we see this. So I see someone in the chat is talking about the sound. Everton, is it okay from where you are?
Here it's okay. I don't know if.
Okay, so it's not on my end at this particular moment.
There is a bit of noise, but it's at least clear to me. I don't know if for everyone else it is.
Okay. Yeah, maybe do say on the end on the chat if the sound is going bad, because I know that my internet connection is going a bit wobbly. So okay, so I'll just continue. It looks like it's probably okay. All right, everyone seems to be saying it's okay for them. All right, cool. Thanks, everyone. Right, okay, so in addition to finding that these distributions that we're finding are matching the experimental data, we also found that giving heterogeneity makes the learning more robust. And it makes it more robust in the sense that if you mistune some hyperparameters, if you have heterogeneity, it doesn't matter so much. What do I mean by that? Well, typically in modeling, you often find that people set up some architecture with some number of neurons and some learning rate and various things like that. And they have to tune that in order to make the network learn the task well. You have to find very particular values of all of those constants. And somehow that's not very realistic if you think about the sort of tasks that real animals would actually face, because they don't know exactly what environment they're going to come into. We can still learn quite generically tasks that surely nobody has ever seen before. So we have a lot of robustness. Most animals have a lot of robustness, and yet, yeah. And so whatever our hyperparameters are, they don't need to be too highly tuned, and heterogeneity seems to help with that. All right. So what's our conclusion from that? Well, the conclusion is that heterogeneity is a metabolically efficient way to improve performance and make learning more robust. What do I mean by metabolically efficient? I mean that for a brain, it doesn't cost anything to have heterogeneity, right? In fact, quite possibly the opposite. It may be the case that it would cost you something to try and engineer to construct every neuron having precisely the same time constant. So you get heterogeneity more or less for free, and it gives you a benefit in terms of performance and learning. And almost without having done the experiments, you'd kind of think, okay, well, it probably uses it then in that case. Like if you have something that costs you nothing and gives you some benefit, then surely the brain is going to be making use of that. All right, so that's the short version of the talk. Feel free to switch your brain off now if that's enough for you. If not, let's dive into some of the details. Okay, so let's start with the methods and the architecture of the model. So in every case, what we're looking at is tasks where you have to classify a stimulus represented as a pattern of input spikes. So before we start, we've always converted like the image or sound or whatever into some pattern of input spikes. And you need to guess what the class of those input spikes are. Okay. And we're doing that with spiking neural networks. So we take those input spikes, we feed that into an input layer. That input layer is then connected into a recurrent layer of leaky integrated fine neurons, which are then connected to a readout layer, which we then use to generate the prediction of what the class of the stimulus should be. All right, so this is actually extremely simple. In fact, I'd say that this is just about the simplest possible architecture you could have that could feasibly do this job. And not only is it a simple architecture, typically in most of our runs, I think we have something like 100 neurons in this recurrent layer. So it's really small, very simple networks. What we're not doing is aiming at like winning machine learning benchmarks, right? We're not aiming to get like the absolute state-of-the-art performance in any of these tasks. What I want to do is precisely find out, I run the same thing with homogeneous network versus a heterogeneous network. How much do you get from heterogeneity? What is that heterogeneity giving you? It's precisely asking the question about what heterogeneity is giving you, not trying to maximize performance. Okay, and I should also say that we are certainly not the first to use this simplest possible architecture. There are many, many, many papers using such an architecture. All right, so now let's have a look at the training. So we looked at 4 different training conditions. I'll talk through these in a second. In every condition, when we're training on the task, the synaptic weights can always be learned. So in terms of this diagram here, the thickness or color of the arrows is telling you the synaptic weight. So you can see that at the beginning of the training, these are the weights, and at the end of the training, they're different. So this dark one here has become light over here. This light one here has become dark over here. And what differs between the 4 conditions that we did training in is the extent to which time constants can be learned, essentially. So in the first condition, in the blue box here, I'm going to use a consistent color scheme for the different training conditions throughout, but you don't have to memorize it. It should be there on every slide. So in this one, we initialize every neuron with the same time constant. So this is the fully homogeneous case. Every time constant is the same, and I'm representing that with the color of these nodes in this graph here. And they can't change throughout time. They can't change during the training. So at the end, they're the same color as they started with. All the time constants haven't changed. So this is the fully homogeneous case. We can introduce a bit of heterogeneity into this by initializing them all with random time constants. So here you can see that the colors are all different, but we don't allow them to train. So whatever value they have at the start is the same that they'll have at the end. Okay, so that's heterogeneous initialization, but a standard version of the training. Only the weights get trained. Or we can start with the same network, in this case, all of the neurons having the same time constant, and we allow those time constants to change during the training. Okay, so they start all the same, but they can end up being different time constants at the end. And then the fourth case is we start them with one set of random values and they end with a different set of random values because they can change. So that's the fully heterogeneous, fully homogeneous, and then 2 different intermediate versions there. Okay,
then, before I continue, can I ask you something? These connections that you're training, are they positive and negative? Do you have any specificity there?
Yeah, we decided not to try and model excitatory versus inhibitory neurons. So they're allowed to switch between being excitatory and inhibitory. Actually, there was an interesting paper from Blake Richards that just came out a few days ago showing how you can train artificial neural networks easily to have excitatory versus inhibitory neurons. When we did this, that was a tricky thing to do, so we just allowed them to go both positive and negative.
Okay, thanks. So there is also a question in the chat here. It's how are the initialized homogenized time constants related to the size of the window of the input spike pattern?
Yeah, I'm not 100% sure what the question means, but
how the time constants are related to the input pattern, so the timing of the input pattern.
Yeah, I think so. So we chose the homogeneous time constant based on a grid search to be the one that gives the best performance. And I think for all of our tasks, it was about 20 millisecond time constant. And the stimuli tended to be about 200 to 300 milliseconds long. So it's about 1/10 of the length of the input spike patterns.
No,
if that's the question.
Yeah, I think that's it. Venkat can confirm, but okay, thanks.
Okay, cool. All right, so let me continue talking about the training. So as before, it's a supervised learning task in the sense that we get an input pattern and then there is a correct answer and the network gets feedback about what the correct answer was. And so the standard way to train these things, if you're using artificial neural networks, is to use a gradient descent, and it's a current neural network, so you want to use backpropagation through time in order to do that. And there's very nice methods to do that, and you know, if you know all about like deep learning and everything, you know that this is an extremely powerful method to train these things. Now, the problem we have with the spiking neural network is that this is called gradient descent because it relies on the existence of gradients. And you only have gradients if things are differentiable. The problem is that spiking neural networks have an essential non-differentiable element, which is that they fire a spike after a value crosses a threshold. So spiking neural networks are not differentiable in this sense, and so you can't directly use gradient descent methods. So there are some solutions to this. The solution that we used is one from Friedemann Zenker and Emre Nefsi called surrogate gradient descent. I probably won't say a lot about how this works because it's not my work, but I think it's really cool. I would definitely encourage you to go and have a look at it. The basic idea is that you treat the network differently in the forward and the backward pass. So in the forward pass, you treat it as a standard spiking neural network with a non-differentiable bit. But in the backwards pass, when you're computing the gradient, you pretend that the spiking non-differentiable bit is in fact differentiable, and you smooth it out so it makes it differentiable. And you're optimizing essentially a different function from the one that you actually want to be optimized. But despite that, it works. And it trains spiking neural networks to perform very well at a very wide range of tasks. So it's kind of a bit of magic somehow, but this thing works. And it's very general, and we've been using it a lot lately. If you want to know more about that, Friedemann made a tutorial with source code to do it called SpyTorch, which I think is pretty cool. And if you Google SpyTorch, you'll find that. And I'd really encourage everyone to look at that. I also wanted to mention that we had a workshop on a whole bunch of people who are using, coming up with new methods. Surrogate gradient descent is just one of several that have been invented lately for training spiking neural networks. And that was a really cool meeting. And we actually wrote up a sort of mini review based on that workshop. And that's going to be coming soon, either in February or possibly in March in Neuron. So keep an eye out for that coming up soon. It's exciting times for spiking neural networks, I think. All right, so back to what I was doing. Let's have a look at the tasks now. So we trained on 5 different tasks with different amounts of temporal structure, and I'll just talk through them all. In general, the ones with less temporal structure I've got over here on the left, and the ones with more temporal structure over here on the right. The visual ones tend to have less temporal structure, and the auditory ones tend to have more. And now I'll just quickly talk through all of the 5 tasks that we use, just so that you know a little bit about what they are. So the first one is called MNIST, and that's neuromorphic MNIST. So just in case you don't know what MNIST is, it's a very common machine learning, computer vision task where you have to recognize handwritten digits. So here is 0, for example, and you get given some training ones, and then you have to test on ones that you haven't seen before. And they're fairly small images. I can't remember exactly how many pixels that is, but it's not a large number of pixels. So the N in NMNIST is neuromorphic. And basically, it takes a substatic image and turns it into spike trains. And the way it turns it into spike trains is by using a dynamic vision sensor, DVS, or event camera. And basically, these are cameras that somehow mimic spiking in some way. Basically, they emit what I would think of as a spike or an event when the brightness value of a pixel changes over some threshold. So you can see that here. You've got some sort of blue spikes that's like a positive change, and red spikes being a negative change, or the other way around. I can't remember which. In particular, for NMNIST, they presented statically the MNIST digits on a screen, and they moved the event camera around to simulate the eye making saccades over this image. So that's how they create a time varying signal from a static image. Okay, and so that's the spikes that we use as input on the NMNIST task. Now, as you might have realized from this, although there is temporal information here in the timing of the spikes, it's not really telling you anything about the structure of the stimulus itself, because the stimulus itself is just a static image. So all the timing information in NMNIST is telling you is about the structure of the saccades that the camera is making. Okay, so that's the least temporal information task. The next one is fMNIST, and this is a fashion MNIST. And this is another fairly standard sort of machine learning task. In this case, the tiny images are pictures of things like shoes and handbags and clothes and things like that. And you have to guess what the picture is of. In this case, we didn't use the event camera method. What we used is we assume that each pixel in these images is an input current to a neuron. And therefore, the brighter the pixel, the higher the input current, the earlier the neuron fires a spike. So that's the way you get timing in this particular task we used. And we just used that because that's something that's been used by a number of papers training spiking neural networks to do tasks lately. But again, although you have timing in the input spikes that you get from this, it's still a static image that is the underlying stimulus. So there's still not much temporal structure here. Okay, so the next task we used is got some real temporal structure, and it's also using one of these DVS event cameras. But in this case, it's a task that has real temporal structure. So you place the camera and someone stands in front of it and does gestures like waving clockwise or waving anti-clockwise or different gestures like that. And you have to guess from the pixels. And I hope you can see these. You can see some of the pixels here on this. I hope the resolution is good enough that you get a view of that. That there's a sort of hand waving around there. So in this one, the spike timing and location really has actual information about what the gesture is. But actually, it turned out that even this one doesn't have as much temporal structure as we would have liked, in the sense that if you just average all of the frames of these, you can actually do fairly well at guessing what the gesture is, even if you discard all of the temporal information that is actually available in the stimulus. So this temporal information does give you some information, and the temporal information does carry some useful information about what the stimulus is, but it turns out that you can do fairly well without using it. All right, and then we had two more or less the same auditory tasks, and they both are spikes that come from a fairly detailed model of the cochlear nucleus. The cochlear nucleus is somehow the second layer in the auditory system. You have the sound comes into the cochlea, goes to the auditory nerve fiber. That's the first sort of layer of the auditory nerve. That's the first layer of spiking neurons. And then the second layer is the cochlear nucleus. And so these are spikes from a model of the cochlear nucleus in response to different sounds. And you sort of have frequency on the y-axis here and time on the x-axis. And there are two sets of stimuli. In the SHD dataset, that's the spiking Heidelberg digits. It's people reading out things like 1, 2, 3, 4, 5, 6. So it's digits. It's the equivalent of MNIST, but for auditory. In the SSC, that's spiking speech commands. So speech commands is things like go, stop, things like that. So it's the speech commands dataset put through the same model. And this one has fewer classes than this one. So SSC is a more difficult version. Okay, and these auditory tasks have quite a lot of temporal structure. Okay, all right, good. Let's continue. So that's basically it for the methods. I'm going to go and talk now about the performance, the results. Okay, so sorry, this slide is a bit of a dense one. I'll talk you through it a little bit slowly. So what we have here is a table of how well it does in terms of performance. And as I said before, we've got these four conditions. So homogeneous initialization, standard training, remember that's just training the weights and not the time constants. Heterogeneous initialization, standard training, or heterogeneous training, that's these two down here. Okay, and you can see that on this key here as well. So the top line is the baseline. This is the free homogeneous network, right? Every neuron has the same time constant. And we don't change the time constants during training. So that's our baseline that we want to compare to. And we've got the 5 different tasks here. And this is chance level, just to give you an indication of that. Okay, so let's go through the tasks and see. So first of all, we have the NMNIST. And kind of as you might expect, given that there isn't much temporal structure here, you don't see any improvement from having heterogeneity at all. It basically gives the same performance regardless. Next, we move on to FMNIST, which has a bit of temporal structure, but not much. And here it's a little bit difficult to interpret. I've got another picture I would like to show in a second. But basically what you see is that heterogeneous initialization helps, but the ability to train the time constants doesn't really help. And you can see it gives you about an 8% improvement, the heterogeneous initialization. So actually, I think this is slightly misleading, and I would like to find a better way of presenting this. But I think basically what is happening actually is that in the homogeneous initialization, the training is getting stuck on a local minimum, but not every time. So here you can see that this is a plus or minus 7.4. And this is about 7.4-ish higher than this number. And basically, if you look at the training curves for the FMNIST, I'll show you in a second, it basically looks like what's happening is sometimes this is landing at a low value, and sometimes it's landing at a large value. Whereas if you initialize with the heterogeneous initialization, it's always getting the best value. So it looks like in this case, what the heterogeneity is doing is helping you not get stuck in a local minimum. And I've got a PDF which I will show you. Zoom in here. So this is the training curves for FMNIST. And you can see here that when your initialization is homogeneous, that's the blue and the green case, you have this wide range of values that you could end up in. Whereas if your initialization is heterogeneous, you have very narrow distributions. But the best values that you can get to with the homogeneous initialization are just as high as the best values you could get to with the heterogeneous initialization. So I think that that's what's going on in that case. All right. All right, so now we move to the DVS data set where you have real temporal information that actually is helping. And you can see here that each form of heterogeneity is giving a little bit of advantage compared to the baseline. And essentially, the more heterogeneity you have, the more advantage you're getting. But the amounts are still relatively modest, so going up from about 77% to about 82%. All right, so now if we move to the auditory ones, which are the ones where they have the greatest degree of temporal structure, these two are the auditory ones, remember, you can see we get a big improvement. So you can see we jump about 10-ish points, 10 percentage points in terms of performance increase from the baseline to the fully heterogeneous case. And that's true for both of these data sets. You also see that, by the way, these are the testing accuracy curves during training. So you can see here that the homogeneous ones are a little bit ahead for DVS, but they really jump ahead a lot for the auditory ones. All right, okay, so Jonathan is asking, how do we train the time constant of the neurons? So basically, we're training the whole network with essentially gradient descent. It's surrogate gradient descent, but let's just say it's gradient descent. What most people are doing when they do that is only differentiating with respect to the weights, but you can also differentiate with respect to any other parameter of the model. And in this case, we differentiate with respect also to the time constants, and then move in the direction of the gradient there. So it's also using gradient descent. Okay, all right. Yeah, but that's all I had to say about the improvement in performance here. Sorry, yes, all of these are accuracies on the test set. Yeah, not on the training set. Okay, yes, yeah, I'll continue, I think. All right, so now I mentioned that the distribution of time constants that we get sort of matches what you'd see in experiments. And here's a little bit more detail on that. On the right here, you've got experiments, and on the left is the simulations. There's a lot of information here, so I'll talk you through this somewhat slowly. So the top row here is NMNIST, then we've got FMNIST, we've got the DVS, that's the gestures one, and we've got one of the two auditory. We've only just shown one of the two auditory, and not both of them because they're fairly similar, and just to try and make this graph a bit more readable. So what we have on the top of each of these histograms is what you get when you do a heterogeneous initialization of time constants. So this is the distribution of time constants that we initialize with. And by chance, we chose a gamma distribution, just because it's a nice distribution that's always positive. That it was purely a guess, actually, on my part. It actually looks quite like the trained distribution, I think. It's a fairly decent fit for it. So that turned out to be quite a good guess, but it was just a guess. And on the bottom is the histogram when you have initialization all being the same. Okay, so it's just each one. So the initial distribution is the same for on the left-hand side. This is the before training distributions. And this is what you get after training. So the red is the distribution of time constants you get after training if you started with the gamma distributed initial distribution. And the green at the bottom is the distribution of time constants you get if you started with a single homogeneous value as the initial distribution. So orange goes to red and blue goes to green, essentially. Okay, so already you see one interesting thing here, which is that in most of these cases, the final distribution that you get to is the same regardless of whether you started off with a heterogeneous or started off with a homogeneous distribution. And so that's nice. That gives you some sort of suggestion that maybe we're finding something that is at least fairly close to the optimal value of the distribution of these time constants. It's not entirely true. Like, there are some slight differences, and you can see here, particularly for the DVS, it looks like there's probably a couple of different solutions in the sort of space of distributions that you could arrive at. And this one has arrived at this, but the shape, even though the mean is slightly shifted, the shape is still fairly similar. And not only that, but these shapes, which I say still actually fit fairly well with either a log normal or a gamma distribution, also look like what you see in real data when you measure these. So at the top here, we have something from the cochlear nucleus of a mouse. That's from Paul Mannis's lab, recordings over many years, released in a paper a couple of years ago, I think. So that's auditory. Then we have a mouse visual one, putatively excitatory neurons. And this comes from the Allen Institute. And by the way, if you don't know about the Allen Institute database, you really should. It's absolutely spectacular. You have thousands and thousands of neurons with huge numbers of properties recorded from it, huge numbers of stimuli that they've shown these animals, and it's really an incredibly rich data set, freely available, and with a Python API that lets you just download these. And we even have the Allen Institute even did some human neurons. So here's what this distribution looks like for some human MTG neurons. Okay, so what we found looking at the data is you tend to see the same pattern for everything we looked at. If you isolate a particular area and a particular neuron type, then you'll see a distribution like this. If you have just one area, you don't see this distribution because, for example, inhibitory maybe would be a curve that looks the same but over here. So you'd have a double peaked distribution because the inhibitory ones would have longer time constants, for example. So you do have to narrow it down to a particular area in a particular neuron type to see these curves in the experimental data. But if you do that, and if you've got enough neurons that you can plot one of these histograms, then it tends to look like this, more or less. All right, there's a couple of questions I see. Did you try other initializations for the distribution of the time constants? We haven't done a lot, mostly because this training is very intensive. I didn't mention this, but the total training time for this paper we estimated was about 2 years of GPU time. So each extra thing that we do really costs a lot in terms of training time. So we haven't done many distributions. Informally, yes, we have tried other distributions, and it seems as though it tends to look like this. I mean, like, as you see, they're not exactly the same every time, but they tend to be roughly having this somewhat gamma or log normal-like distribution, is what I would say for that. Is the distribution of heterogeneous time constants a hyperparameter and tuned? Actually, the answer for that is no. We used the same mean as the homogeneous, but we just picked one particular value for this heterogeneous distribution of initial time constants. Venkat, I think I'll answer your question at the end if we have time, because I think that's a bit more speculative. And I'll just continue now because I realize actually I'm actually ending up using quite a lot of time in this talk. So I'll keep going for the moment and come back to your question at the end, Venkat. Do you want to ask it at the bottom so that we don't miss it? Okay, right. So yes, I think that was all I wanted to say about that. All right, now, so the next result is about robustness. So I mentioned one sort of robustness already earlier, which was in the FMNIST example. We saw that it seems to let you break out of, well, maybe I should just show this PDF again. Basically, if you have the homogeneous initialization, you get a wide range of possible training. So you might end up with a good result, but you might end up with quite a bad result as well. Whereas with the heterogeneous one, you sort of break out of those local minimum. So that's one sort of robustness, sorry. We also see some other sorts of robustness. Oh. Sorry, my screen's doing something weird right now. Okay, I'll just have to live with that. Okay, that's fixed. Yeah, so what was I saying? Right, yeah, so that's one sort of robustness. We also see some other forms of robustness for the surrogate gradient descent that I haven't talked about. I won't talk about now because actually we're running out of time, and also I wanted to keep this simple. As well as finding robustness in having this heterogeneity, we also wanted to test that the results that we found are not specific to the method that we used, this surrogate gradient descent. So in order to test that, we looked at a different training method to show that we also see an advantage to heterogeneity and that also we see robustness in that. So I'll just talk about that one just to give you another point of view on this. So the method that we used for this one, it's not the architecture I used before. It's Claudia Klopath and Wilton Nicola came up with an update to the force method to make it work for spiking neural networks. That was a good few years ago now. And the task is that you need to get something weird is going on. Okay, that seems to be back again. And the task is that you need to reproduce a spike train input. Essentially, you have to learn an input spike train, and you then need to reproduce that back again. In this case, that input spike train is representing the spectrogram of, well, in this case, it's a zebra-finch pool. So it's just a stimulus that has some rich temporal structure and also some spatial structure as well that you need to learn. All right. So in their work, what they did is they had, I think it was 1,000 neurons. They all had the same time constants. And so in the same way, we decided, well, we could also make those time constants heterogeneous instead of all of the same. What effect would that have? So to look at the effect, I wanted to talk about hyperparameter mistuning. So what they did is there are let's say there's three key variables of their model: the number of neurons n, which in theirs was 1,000, and there are two learning parameters called g and q. I won't get into what those mean, but they're just hyperparameters of the learning. And what they did is they fixed their value at n equals 1,000, and they found the best values of g and q to train the neurons, the network, to do this task. So what we wanted to know was, how well would it work if you didn't do that tuning of g and q? And would heterogeneity help with that? So here's some results for that. So you have here is what they did in their paper. You have n equals 1,000. On the axes here are g and q, and basically they picked this value here roughly, which is the value that gives the best performance for all of the different values of g and q looked at for the particular task, this particular number of neurons. And what you're seeing here is how well the color represents how well the network learned to do the task, where basically dark blue is the best, lighter blue is slightly worse, but still okay. And then when you switch into the sort of shades of white and gray, that's when the network has basically completely failed to learn to do this task. So what we immediately see is if you just change the number of neurons a bit, suddenly this network starts failing to learn the task. And already by 3,000, 4,000 neurons, it's really in most of the values of G and Q failing to learn this task. So the first thing we did was to just try introducing a minimal amount of heterogeneity. Instead of having a single time constant, we randomly assign each neuron to have one of two time constants. That's this double case. And already that expands the zone of hyperparameters in which the network can learn the task. Right, so it's already become more robust to mistuning of the hyperparameters by just introducing two time constants. Now, if we spread that out even more and we jump again to our gamma distribution, you can see that that massively increases it. Now, every neuron can have a different time constant and they're randomly selected according to the gamma distribution. Now you can see you can get a long way away, and even here that you start seeing a few failures right in the edge here. But it's become a lot more robust to these hyperparameters being mistuned. Right, so basically the conclusion is from what we've seen is that yes, the heterogeneity does give you robustness, and it doesn't even depend on the training method. It seems to be a general feature of heterogeneity. Okay, all right, so time is running out, so I'm going to move on now to our discussion and conclusions. So my first sort of conclusion point here was, I said before, that heterogeneity is metabolically or energetically efficient. So metabolically in the sense that, as I said before, it doesn't cost the brain anything to have heterogeneity, and so therefore it's a good idea. Energetically efficient relates to something that I didn't show, but to give you an idea, we looked at the auditory digits task. To get the same performance increase that we got using heterogeneity, you'd need to use about 10 times as many homogeneous neurons. So you can get that level of performance if you up the number of neurons. But that increases the number of parameters by 100 times, right? Because it's a fully connected network, so it squares the number of parameters. Whereas putting heterogeneity in increases the number of parameters by less than 1%. So adding heterogeneity, you get the same performance gain with less than 1% extra parameters that you would get by multiplying the number of parameters by 100. So it's sort of energetically efficient in that sense as well. It's a good way to get more performance for less energy cost. And this also spreads to engineering applications as well. If you think about it, the cost of introducing heterogeneity is somehow O of n compared to O of n squared for adding more neurons. If you're thinking about neuromorphic computing, you may even get it for free, like you get with the brain. For example, in the Brainscales architecture, they actually manufacture the hardware so that each neuron is heterogeneous because they can't avoid having heterogeneity, and so therefore they get this heterogeneity for free actually as well. And also, as I said before, having this robustness to hyperparameter mistuning is probably important in the real world. Okay, so to answer the questions that I asked at the beginning, yes, sometimes we do need to include heterogeneity. Obviously, it depends what we're studying. You don't necessarily need to introduce heterogeneity for everything. I'm not saying that every neuron that has, every study that has all neurons being the same has to be thrown out. It doesn't matter for every task. But at least we can't assume that it's irrelevant. And for the sorts of things that I'm interested in, which is sort of robust modeling of complex information processing, it's probably pretty important. So at least for those sorts of things, and particularly say in the auditory system, it seems like this is likely to be an important thing. There's a lot of open questions. We've shown that, that heterogeneity can be useful, but we don't know if the brain is optimizing for this or if it's really using it in any sense at all. We've just shown that it could. I think it's likely that it makes use of this, because why wouldn't it? But it's still open whether it really does, I guess. There are other forms of heterogeneity. We've only looked at time constant heterogeneity, but there's many other forms of heterogeneity you could look at. And finally, we would like to find a rigorous theoretical explanation as to what heterogeneity is giving you here. At the moment, this is a pure simulation study. So those are three sort of open questions, I think, for this work. Right, so I'm pretty much done there. I just want to say thank you to Nicholas and Vincent, who are the students who worked on this. Nicholas is my PhD student, and Vincent is supervised by Pierluigi Ducati. I also want to say thank you to Paul Mannis and the Allen Institute for the amazing data. I've already waxed lyrical about that, so I guess I don't need to say more about that. But yeah, this open data thing that's happening at the moment is just spectacular, I think. I think it really changes the way science is done. And for people like me, for pure theory labs, this really is game-changing, I think, in that we can just instantly have access to huge amounts of experimental data to test our ideas out on. And again, I also want to thank Friedemann and Ray for their work on surrogate gradient descent, which is now basically everything I'm doing in my group uses this. So again, game-changing for spiking neural networks, I think. And thank you all for listening. Thank you.
Thank you very much, Dan. It was really interesting talk. It's nice to see how heterogeneity can play an important role in learning. So we have questions here for the end. So I'll try to invite them at the order that appears here. If they want, let's see if I can do this. The first one is from, I have the email here. So P. Miller. Let's see. I think it's Paul. Let's.
Just grab myself a glass of water. One sec.
Okay.
Hi, it's Paul Miller. I was noticing.
Sorry, Paul. I think he went to grab a glass of water.
No, I'm back.
Oh, you're here. Okay, sorry.
Okay. Hi, great talk. I certainly love the idea of heterogeneity, and I noticed though clearly you're focusing on time constants, but time constants aren't kind of like a baseline parameter of a neuron. They're kind of derived from all the conductances plus the capacitance and so on, and also they're voltage dependent. They change with inputs and so on. And I suspect you're using leak integrate and fire, which just has a fixed time constants. So I imagine things would be very different if you had, all the time varying conductances. And I guess
I think it would be really interesting to look at that. Yeah.
Just with the Allen Institute, they must fix a certain voltage membrane potential to measure a time constant, and I assume they will change depending on what voltage you measure it at, because so many conductances vary with voltage.
Yeah, indeed. I can't remember off the top of my head. Maybe someone in chat will know how the Allen Institute measured their time constants. Indeed, they only give one number, so indeed it must be some fixed thing. But I don't remember off the top of my head what the technique was. It's certainly listed in their protocol, but I can't remember what that was. Yeah, I think, so you're right. It's leaky integrate and fire neurons, so indeed it only has a fixed time constant. I think it would be really interesting to look at other properties, a more biophysically detailed model that has conductances and so forth. And I think that it would be very easy to adapt our methods to do that. Basically, you just have to change like 5 lines in our code and rerun it, essentially. You also need a fairly large amount of computer time to do it. But other than that, the thinking time is straightforward. Some other groups have looked at other forms of heterogeneity. For example, another group introduced heterogeneity in the properties of, they used a sort of more complex form of adaptation in their integrate and fire neurons. So it was a 2 or 3 parameter neuron model that had adaptation. And having heterogeneity in that helped even more. That came out while we were doing this, so we didn't include that. But yes, I think the answer is that other forms of heterogeneity is certainly going to help, and they'll be interesting to look at.
Okay, thank you.
Yeah, thank you, Paul, for the question. And Venkat had a question. Can you? I'm unmuting you, and I'm going to release Paul then.
It looks like he's frozen, at least on my end.
So I think I'm going to mute him and go to Bill then. So, Bill?
Hey, can you hear me?
Yeah.
Yes.
Okay. So thanks for the really interesting talk.
Thank you.
So I was wondering, I guess I have 2 related questions. One of them is, did you try to play with the relative learning rates of time constants versus weights to see if, say, learning at a slower rate, the time constants would help?
No, that's a cool idea. Yeah, we didn't look at that. We had it. We played a little bit with learning rates, and didn't make sort of huge amounts of difference. But we didn't try relative learning rates of time constants versus weights. That's a really cool idea. Yeah, it would make sense if you wanted to learn those a bit slower, for example.
Mm-hmm. And then,
that's a nice idea.
And then similarly, like sort of similarly, I'm wondering about the biological plausibility of it. Do you think it would be it's like a very difficult problem for neurons to learn their time constants? Is it somehow easier than the weights? Do you have any ideas about that?
So what I'm thinking of is that I'm not sort of promoting the idea that that time constant learning is necessarily a thing. I would imagine that this is finding what distribution of time constants would be best for the task. So it's kind of like merging like online learning during a lifetime and sort of evolutionary finding that the optimal distribution. However, I think that there is some work on the biological plausibility of like learning time constants and things like that. So it's not totally impossible. At the crudest level, I guess it would probably happen to some extent if you had a larger pool of neurons, in that it would select the neurons with the right time constants, if nothing else, to use in its computations, the ones that worked best. We couldn't rely on that in ours because we only had 100 neurons, so we can't rely on there being a massive pool of different time constants for what we're doing. But you might see something similar there as well. Yeah.
Thanks.
That's not really a complete answer to your question, but we didn't really have a complete answer to the question. So. Okay.
Thanks, Bill. I'm going to unmute Venkat now, if it's working. Let's see.
He moved for a little bit and then his video froze again.
Yeah.
Oh, he's back. He's back. Oh, and now we don't hear you, Venkat, or at least I don't hear you.
Okay, I'm going to mute him again. Maybe I will read his question later. Let's go to Rory Byrne. Can you ask your question, please? Yeah.
Hi. Yeah. So my question is kind of speculative, I guess, but you mentioned, the possibilities of exploring other kinds of heterogeneity. What might those be? Which ones are you kind of most interested in?
Well, yeah. So I mentioned one in my answer to the last question, which was they had heterogeneity of the parameters of the adaptivity, for example. I think it would also be interesting to introduce more cell types into what I'm looking at. So these are all undifferentiated in terms of their cell type. And as I said, I think in respect to your question earlier, they're allowed to switch from excitation to inhibition, which was a decision we made because it was technically more feasible to do that. That seems to have been solved now with Blake Richards' paper, although I haven't yet had a chance to look at that in detail. But I guess that any set of parameters that are relevant could start to be made heterogeneous, I think. I mean, I think anything like that could be relevant. Yeah. So any parameter, yeah. So I mean, in a leaky integrated fire neuron, you don't have that many parameters, right? You've got time constant, and you've maybe got the distance between threshold and reset, and that's about all you've got. Right. But it's more complex, biophysically detailed models you have more to play with.
Mm-hmm. Is it important that it's something that you can differentiate and get a gradient with respect to that parameter, or? Yeah. Mm-hmm.
But that's usually true. I mean,
right.
If it's a model that's set up as a differential equation, that would usually be true, right?
Great. Thanks.
Yeah. Okay.
Thank you, Rory. Let's see if Venkat can join us now. Is it working?
We can see you moving, but don't hear you, Venkat, this time. Maybe you should just read this question.
Yeah. Okay, I'm going to unmute Peter. And then
he can be on the screen and like gesticulate while you read the question.
Okay, I need to find the question. Let's hear Peter, maybe. I unmuted you.
Yes, can you hear me?
Yes, I can.
Marvelous. Nice talk. So I was wondering about synaptic time constants.
Right, yeah, so I didn't talk...
synaptic currents or ideally conductances, which are more difficult, obviously, to implement in the leaky integrate and fire model. So do you think they have an even stronger beneficial effect because you may vary them individually for the synapses converging on the same neuron?
Yeah, it's a good question. So we actually did allow synaptic time constants to vary in this, but for technical reasons, they have to be the same for all incoming synapses to a given neuron. Basically, the way we set this up for it to be computationally efficient, we couldn't have it be different for each synapse. So given that requirement, we don't see any extra benefit from synaptic time constant heterogeneity. But I think you're right. If you allow that to vary per synapse, that would probably help. But we haven't implemented it just because it's just really technically challenging to do that in an efficient way.
Okay, thank you.
And I was muted, so okay. Thank you, Peter. So one last try, Venkat, and then let's see. Then I read your question.
No, still quiet.
No, it's still quiet. So I'm going to read it. So what he wrote here is, intuitively, why don't time constants kind of become big, especially for temporal heavy tasks?
So, I mean, if they're big, I don't see why they would just all go big, right? I mean, if they're large, they're doing integration of information, which is useful for some tasks, but but less useful if coincidence, for example, is important, and then you want them to be small. So when you have information that is present at multiple timescales, you probably want a mix of constants, right? My intuition wouldn't be that they would all go large, possibly unless it was an entirely static, like Poisson distributed spike train where only the rate mattered, in which case you might expect them to go large. Maybe it would be worth checking that. And why the average time constant stuck at about 20 milliseconds? Well, they could be because we did this grid search to find the optimal single time constant. It could be that's why the averages stayed around there. Yeah.
Okay.
Sorry, I was responding to Venkat's follow-up in the chat there.
Yeah, no, I thought. Yeah, thanks for doing that. Yeah, then so now I have Paul here. I'm going to unmute you. And
yeah. Thank you for the nice talk. I was wondering kind of what kind of ansatz you have for the last question that you raised. What is the theoretical reason for the kind of advantage of this heterogeneity, and whether there's anything known from artificial neural networks, whether there are also kind of implemented heterogenesis that help learning. So whether there's something that we can learn from this artificial neural networks in this regard.
Yeah. So Nicholas, my student, might want to say something about that. I don't know if he wants to come on screen. I see he's there, but I mean, I don't immediately have a good idea as to what that theoretical insight would be. A couple of directions that occur to me. One, this is a bit vague, but just thinking about like the advantages that you see in sort of machine learning methods of like random projection methods, for example. They tend to turn out to be very powerful, and there are theoretical reasons why these random projection methods are good. And it may be that heterogeneity is providing something similar to that. If you thought about it in the right space or whatever, you might think that this is somehow a bit like a random projection method. So there may be some mileage in that. Alternatively, it's just that if there is information that is somehow modulating at different temporal scales in the stimulus, then having different temporal scales in your network reflecting that would also help. But that would be very specific to somehow the temporal stuff. That wouldn't be as generic as general for heterogeneity more generally. So those are the sort of two initial ideas I would have for that. But yeah, it's a bit impressionistic at the moment, a bit vague. If you have a good idea, I'd love to know.
Thank you.
I'm open for collaborations if anyone has a good theory idea for this.
Thanks, Paul. So we have other 2 questions here. Let's say that I cannot invite. So I don't know if you answered this yet, already, Dan, but the membrane time constants distributions that are calculated have much longer tails towards longer times. Do you understand this difference? It's from Henry Greenside.
Okay, hang on, let me just go back to that slide. So I think that they're probably saying that the ones that are the
maybe the
options that we find are more long-tailed than our input distribution. Yeah. Well, I mean, our input distribution is, or is the question that it's more to do, they're more heavy-tailed than you see in the experimental data? It might just be that you're not seeing it in the experimental data because there weren't enough neurons. In each of these plots, there's only a couple of hundred, so it may just be that we're not seeing much of the tail, whereas we're averaging over more neurons than that in this that we're seeing it there. Mm-hmm. There's no particular reason it ought to be like our input distribution, just because we just picked this input distribution out of the hat. So there's no reason why it should look the same. Yeah. Does that?
I think, and I have a quick question now in this line, just that you have the slide there again. So for the bottom case, you have these 2 bars, the red and green at the far right, or on around 100 milliseconds, right? So I imagine
yes.
you have
right.
You have some limit there that you impose.
Yeah, exactly.
So
we clip at 100 milliseconds. So it's basically it wants to use longer than 100 milliseconds. Some neurons with more than 100 milliseconds, but we're clipping it at 100.
Mm-hmm. And did you try releasing this clip, or if it helps somehow? Or?
Yes, we did, and I don't remember why we didn't allow it to go arbitrarily large. Maybe Nicholas will remember and can reply in the chat. I think we may have ended up with some instabilities if we let it go arbitrarily large. But I don't think that's necessarily that related to what really is optimal. It's just that the training method was doing something a bit weird when we allowed it to do that, I think. But maybe Nicholas will answer in the chat.
Okay, or you can also continue this. So I'm going to put the link again shortly for our Zoom meeting to continue.
Oh yeah, he's answering.
Okay, and so there is one last question here that I think it's from Tim.
Oh no, he's answering a different question. Okay.
Okay. So I think he posted.
We seem to have lost Everton.
Lost? Sorry.
Have I disconnected?
Oh, I can hear you. Hello. So Dan, can you hear me? Okay, so let's continue maybe our discussions in this Zoom channel that I just...
I'm not disconnected because I'm still seeing things coming up in the chat. I think our host is disconnected.
But you cannot hear anything. So I think let's wrap it up here, and then...
Can everyone hear me speaking?
Yes. Wait, I'm going to mute.
Okay.
Oh.
I'm not hearing anyone else speaking, so I don't know if that's the same for anyone else. I'm only seeing myself and the chat. Okay, yeah, let's jump to the Zoom room. Cool. That works for me.
Okay, let me thank again Dan for this really interesting talk, and so let's go to this Zoom meeting. So whoever wants to join us and continue talking to Dan and asking more questions and discussing further, please join us there. I'll be moving there now too. So goodbye. Have a good weekend, everyone.